The Depixus MAGNA One instrument is a powerful new tool for measuring complex interactions between biomolecules. Here we explain how MAGNA One can be used in binding strength mode to measure protein-to-protein interactions. Double-stranded DNA is attached to a paramagnetic bead via a single-stranded DNA handle. A second handle allows the construct to be attached to a flow cell surface. A pair of DNA-tagged proteins are added and bind to complementary base sequences in each handle. The two proteins are now attached at a fixed distance apart along the DNA. Here, the two proteins do not interact. A highly controllable magnet is slowly lowered towards the flow cell surface, and the beads rise in response to the force. The precise vertical position of each bead is tracked. In this case, an abrupt change in bead position occurs as the beads rise to the maximum height allowed by the DNA. A second pair of proteins is tested. They interact strongly. This time, when the magnet is lowered, the bound protein pairs cause the beads to pause at an intermediate height. The increasing force eventually separates the proteins, and the beads continue to rise. This approach can also be used to test the interaction between a protein and an antibody, or even the binding of a small molecule to a protein. An alternative simple experiment can be used to study protein pairs that interact weakly or intermittently. DNA-tagged proteins are loaded into the flow cell as before, but the beads are subjected to only a very low constant force. The vertical component of the bead's Brownian motion shows two levels of amplitude. The amplitude is high when the proteins are not interacting, but is lower during protein binding. This provides information on the interaction kinetics, such as the K-on and K-off values. The effect of small molecule drug candidates can then be assessed. Here, an inhibitor prevents the interaction. Conversely, this molecular glue holds the proteins in the bound state. Depixus MAGNA One. See biology as it really happens.